So I'm talking today about some work that's ongoing in my lab investigating the gut microbiome in Huntington disease. So I got interested in this because pathological weight change is a feature of Huntington disease, with the vast majority of individuals undergoing pathological weight loss and a small minority either maintaining their BMI or in some cases undergoing weight gain. And there's a connection between weight loss and symptom severity in Huntington disease, whereby the rate of functional decline is slower for individuals who have a higher BMI, and the rate of motor decline and the rate of cognitive decline, these are all associated with the BMI. And this suggests that there's some sort of link between the metabolic dysfunction in Huntington disease and the dysfunction in the brain. And so this suggests that there's something disrupted in gut-brain feedback in Huntington disease. And so gut-brain feedback is bidirectional communication between the brain and the gut, and it's ongoing, and it's kind of a chicken and the egg thing. Like if you change the brain, you necessarily change the gut, and if you change the gut, you're going to change the brain. And so we became interested specifically in how changes in the gut microbiome might be affecting the brain in Huntington disease. The microbiome is the collective genome of all of the microorganisms in the gut, and the microorganism cells in an individual not outweigh, but there are more. And I know people say this thing like, we're 90% microorganisms, but we're not really. There are 10 times as many microorganism cells in your body as there are your body cells, but they're very, very small compared to your cells. So even though there's 10 times as many of them, they only make up about 1 to 3% of body mass. And the greatest concentration by far is in the gut, although there is a skin microbiome and an oral microbiome, but by far the most is in the gut. And the microbial DNA far, far outnumbers our DNA, about 100 to 1. And while the microbiome does include lots of different types of microorganisms, bacteria are far and away the most abundant. And we have evolved, we've co-evolved with these bacteria, and these bacteria have obligate functions. There are certain parts of our diet, certain parts of metabolism that we can't perform by ourselves. We rely on the microbes in our gut in order to metabolize all of our food and to synthesize certain amino acids and synthesize certain vitamins. And neurotransmitters are produced in the gut. And so there's a lot of necessary functions that these bugs in the gut play. And when this gets disrupted, it can have effects on the whole organism, and certainly including the brain. And when the brain is speaking to the gut, this primarily goes through the hypothalamus or the vagus nerve. But when the gut is speaking to through the brain, this is often going to be factors that are released from gut microbiota, things like cortisol or pro-inflammatory cytokines, which are chemicals that stimulate the immune system. As I said, there are lots of neurotransmitters, serotonin. There's lots of serotonin that is made in the gut, and then it goes up and it acts in the brain. And fatty acids that are part of the bacterial metabolism. And changes in these systems have been implicated in all kinds of neurological disorders. Increased bacterial cortisol has been linked to depression, and bacterial-derived pro-inflammatory cytokines are known to cause neuroinflammation. Changes in bacterial tryptophan metabolism have been linked to schizophrenia and Alzheimer's disease. And anxiety has been linked to changes in the amounts of neurotransmitters that are produced in the gut. And additionally, there are fermentation byproducts, these fatty acids, that have been linked to autism spectrum disorder. So The changes in the gut and the way that the gut feeds back to the brain have roles in all of these different varieties of neurological disorders. And it's been shown that there is gut dysbiosis in Huntington disease. This study that came out in 2020 was performed in Australia, and this was the first study looking at the composition of gut microbes in Huntington disease gut. And they found that there were alterations, and that those alterations were linked to the symptoms. Another study that was done in North America found a similar thing, that there were changes in composition, but the changes that were identified were different. So this is kind of the classical way that microbiome composition is looked at, where you can look at the actual bacteria themselves, and you can look at them at the phylum or class level, at the family, or down at the genus level. You can even look these days at specific species. So this is looking at the identity of the bacteria. And this is showing here, this is Huntington's disease, and this is control. And if you look at these colors, the different sizes of stripes indicate that there are changes. This whole bar is 100% of the bacteria in the gut. And here in this study, they showed this dark blue here is a genus of bacteria called Bacteroides, sorry. And that was found to be less in the HD gut than in the control gut. But different changes in composition were seen in the two different studies. And they were even more different than that, in that one study found that there was a reduction in diversity of bacterial species in the HD gut, and the other study found that there was an increase in diversity. So the studies both found that the HD gut was altered, but they saw different changes in the composition. But importantly, both of these studies found a link between symptom severity and the changes that they identified. In the Australian study here, they found that changes in Eubacterium hallei, which is a specific species that they identified that was altered, are related to severity of motor signs, and even to the predicted age of onset in pre-manifest gene expansion carriers. And the North American study found links to functional capacity and cognitive ability. So even though they found different changes, they both found that they were important in Huntington disease. And one of the reasons might be that we have these obligate functions for bacteria to produce in our gut, but they don't, like, more than one species of bacteria can provide each function. So say there's a vitamin that needs to be synthesized, there might be 10 different types of bacteria that can synthesize that vitamin. And as long as you have enough of any bacteria that can synthesize that vitamin, then you have a healthy gut. And so this more traditional analysis, where people are studying the composition in the gut, may not be as relevant as a newer kind of analysis that's being done now, looking at the functions. So looking at, there are these obligate functions of bacteria and all of the potential species that could serve that function, and looking and see if there are deficits there. And I feel like that's going to be particularly important for Huntington disease, because there is a dramatic change in metabolism. Now, these individuals that are undergoing rapid pathological weight loss, it's not related to energy expenditure. People are consuming more calories than they are using and still losing weight, which suggests to me that these obligate functions of the gut in helping us to metabolize our diet and make use of it are disrupted. So I'm more interested in these kinds of functional studies rather than looking at specific species, which we've already seen can be very different in different countries. You know, the Australian study versus the North American study had quite different actual species of bacteria that were altered. So one of the ways that the microbiome regulates the brain is through production of metabolites. Bacterial metabolites enter the circulation through the liver, and these can reach or exceed normal drug concentrations, and they can pass through the blood-brain barrier and have very substantial effects in the brain. So when we started thinking about functional analysis, this is where we started. We looked at what metabolites are altered in the circulation of individuals with HD. And this is from a study in 2015, where another group just looked at alterations in any metabolite, not necessarily those produced by bacteria. And so we took their findings of which metabolites in general were altered in HD, and we broke this down to which ones are only produced by bacteria. And we came up with 4 metabolites that are produced by bacteria that are changed in Huntington disease. Now, we didn't pursue these antioxidant metabolites at the bottom here because not enough is known about their synthesis. We don't know of bacteria that inhabit the human gut that produce these. We know that they are only produced by bacteria, and so there must be human gut bacteria that produce them, but we don't know which species of bacteria they are. So it's not a path that we can currently follow. We also decided not to look further at indole-3 lactic acid because it was only found to be altered in pre-Huntington disease, not in Huntington disease itself. So we thought it wasn't as high a priority as indole-3 propionic acid, which is reduced pre-symptomatically, but also in manifest Huntington disease. And next, we identified all of the known bacterial producers of IPA, indole-3 propionic acid, and that are known to inhabit the human gut. And we came up with these 8 different species. It's from 2 different genera. There's Clostridium species, and then there's Pipistreptococcus species. And so this is one way that just looking at composition, this is an example of how just looking at composition wouldn't necessarily tell you if a gut was healthy or not, because you could have any one of these 8 bacterial strains, and as long as you've got something there that's producing enough IPA, it's going to be healthy gut in terms of IPA production. But just looking at the genera or the species won't give you that information. So we looked a little bit further into IPA, and it turns out that it's quite an interesting metabolite. It's an antioxidant, and it has potent antioxidant activity in the brain, which the Huntington disease brain, oxidative stress is something that occurs normally with biological aging, but it's elevated in the Huntington disease brain. So this enhanced oxidative stress is a feature of HD. We also found that the absence of IPA producing bacteria in the gut of animals leads to a hyperactive innate immune response. And this is intriguing because the HD immune system is hyperactive. There is a hyperactive response to immune stimulation that's seen as much as 16 years before onset of motor signs in Huntington disease. So this hyperactive immune system is a very early feature of HD, and it's something that can result from the loss of IPA. Additionally, IPA fortifies the intestinal epithelium. So, ensuring that the lining of the gut is healthy. Now, this is of interest to me because disruptions in this intestinal barrier are known to be linked to mood disorders, including anxiety and depression and impaired social function, and also cognitive dysfunction, which are all features of Huntington disease. So we found IPA to be very intriguing in that it's reduced early in HD, and its loss results in some of the early changes that are seen in HD. Now, that doesn't mean that it's necessarily causative, but this certainly made us want to dig deeper and look further into IPA. So our microbiome study that we have underway, we've got two different arms. One is an untargeted study, and this is the more traditional approach for microbiome studies, where we're quantifying relative abundance of microbial taxa in the HD gut. We are looking both at separately at individuals who are either underweight or undergoing rapid weight loss versus those that are not, because we're hoping to identify differences in the gut that may be responsible for this, or maybe ways that we can modulate weight loss in HD. We're going to look at this in the traditional method of composition, but we're also going to use the newer type of analysis to look at the functions of bacteria and try to identify targets that we may be able to use to make the HD gut healthier. And our second goal of the study, it's a targeted approach. We want to quantify the relative abundance of bacteria that perform these specific functions, starting out with bacteria that produce IPA. And in our untargeted approach, in our functional analysis, we may identify other functional deficits in the HD gut that we can pursue in this targeted approach as well. As I said, we're looking in different populations for Huntington disease. There's healthy controls, but we're also looking at underweight or undergoing unexplained weight loss, normal to overweight individuals, so that we can potentially identify if there are gut changes driving these two different aspects. We're pairing this study with the Enroll HD study, which means that individuals will have undergone a full clinical assessment so that we can look at the symptom severity in HD. We're collecting plasma because we want to look at bacterial metabolites in circulation and see if those tie to changes in the gut, and see if those also link to symptom severity the way that other groups have shown that changes in the gut do. And then we're sending people with a home stool collection kit. And this will allow us to look at the variety of microbes in the gut. This is what our collection kits look like. They go home with people at the end of their enroll visit, and they come with a box that is postage paid. And so people do the collection at home, and then seal up the box and drop it in the mail, and it comes directly to the lab. We've currently got sample and data collection ongoing at 3 different Huntington disease clinics, but we're hoping to expand this. We'd like to have larger numbers of individuals from different geographic areas, because we're most interested in finding changes that are the result of Huntington disease and not necessarily the result of individual variation or geographic variation. Now, since we started doing these collections, some other interesting evidence about IPA producing bacteria came out. There have been a couple of recent studies that, instead of looking at the functions of the metabolite IPA specifically, looked at what happens in animals when you give them a probiotic treatment of a bacteria that's an IPA producer. So this study gave just normal mice Clostridium sporogenes, which is a species of bacteria that is known to be not harmful to people, that is known to inhabit the gut, and that is an IPA producer. And when they gave this probiotic, they did see an increase in this particular species in the gut. So the probiotic actually colonized the gut, and the bacteria stayed there. And they did measure an increase in circulating IPA once they gave this probiotic. And interestingly, they saw an increase in the average daily body weight gain. Now, this is particularly interesting to me because weight gain is not an effect of just IPA, the metabolite itself, but it is an effect of providing a bacterium that is producing IPA. So there's appears to be a functional difference between just taking, IPA, for instance, as a supplement versus having IPA produced locally in the gut in terms of regulating body weight. And another study that also looked at the same species of bacteria found, again, an increase in IPA in circulation after they gave this Clostridium sporogenes. But they also found that this probiotic treatment was able to promote peripheral nerve regeneration. So injury to peripheral nerves happens faster with this probiotic treatment. Now, this is a really interesting finding because everything we knew about IPA previously was about, oxidative stress, anti-inflammatory and gut epithelium. But this suggests that IPA has some specific role in promoting nerve health, neurite outgrowth, the connections between neurons, which is one of the earliest things that goes wrong in an HD brain is loss of connections between neurons. So we decided to start a study looking at how probiotic treatment with Clostridium sporogenes will affect Huntington disease mice. We're using HD and normal mice, giving them this probiotic. And at the end of the study, we're looking at whether the probiotic actually colonized the gut, whether we see an increase in IPA in circulation, if it slows down the body weight loss of these animals. We're looking to see if it can reduce the pro-inflammation in these animals, if it can reduce oxidative stress, but also looking at HD-like behaviors, since there's been this link with nerve health and IPA function. And this study is still ongoing and we haven't collected all of the animals yet, but I have seen in some preliminary data that it does appear to be slowing the body weight loss in these Huntington disease mice. So very preliminary, but encouraging results so far. So in summary, gut flora have a big impact on brain health. And we know that they're altered in HD, and we know that the changes in the gut in HD are related to the symptom severity of HD. So if we can understand these gut changes, we can potentially improve HD gut health and maybe improve HD. So we're focusing on functional analysis because the different bugs in the gut can pinch hit for one another. As long as you've got something performing a particular function, it doesn't matter necessarily exactly which bacteria it is. So we're concentrating on functional analysis, and we're concentrating also on this bacterial IPA. So because it seems to potentially have these roles in inflammation, oxidative stress, weight loss, and maybe even neurological features. Acknowledgements. Casey Hart is the student in my lab who identified IPA as being a bacterial produced metabolite and which bacteria we should be quantifying, and developed methods for quantifying all bacteria that produce IPA. Christian Milot is a student in my lab who is working on the probiotic trial in HD mice. And Robert Shee is my second in command, who is working with me on the clinical study. Also, we have our clinical collaborators who are collecting samples and clinical data for us. Juan Sanchez Ramos at USF, Blair Levitt at the UBC Center for Huntington Disease, and most recently, Anwar Ahmed and Danish Bhatti from the Central Florida Center for Huntington Disease here in Orlando. and also Sarkis Mazmanian at Caltech, who has been consulting with me since microbiome is not really my area of expertise. Are there any questions?
Is there any data on the prior gut microbiome composition, or prior gut microbiome composition influencing colonization success of the probiotic?
So really, there's only the two studies that I showed, the Australian study and the North American study. When we started working on this project, there was not a single HD microbiome study that had ever been done. I became interested in it from my graduate work, working with Sarkis Mazmanian and Paul Patterson. They were some of the folks that identified a link between autism spectrum disorder and the gut, and Sarkis had identified a link between Parkinson's disease and the gut. And it just kind of seemed like every neurological disorder that was studied, there was some sort of link with the gut. And it became more and more obvious how much of brain function and brain health is dependent on this crosstalk from the gut. And so, you know, really all we know, since those studies had such different compositional findings, what we know for sure is that the HD gut is changed and that those changes relate to the symptoms.
Hi, I just wanted to ask a quick question regarding the studies you showed. I think the findings are rather interesting, particularly about this Clostridia, and that it can help potentially increase weight in your mice and maybe help neuronal survival, although at least, I guess the question is, you showed data on peripheral nerves, and we don't know whether that's going to help the CNS nerves. So
right. So that paper came out a couple weeks ago in Nature. So it's a very, very new finding. We had been pursuing IPA because of the antioxidant and anti-inflammatory roles, and we knew that those were important in HD. And we were very excited when this Nature paper came out. So no, nobody has looked at the CNS yet. But we do know that IPA does cross the BBB and act in the brain.
Wow.
So,
do you know, have you kind of examined why are your mice gaining weight? Is it because they're just eating more? Does their bowel pattern change with treatment?
we do not know. You know, that's all that stuff that we want to look at in the future. One of the things that we've found in our HD mouse models is that they have disrupted feeding regulation. So they don't have the kind of normal circadian rhythm of feeding, the feeding pattern that normal mice do. They kind of eat at random times. And we found that artificially restoring their circadian feeding pattern by only offering them food when they're supposed to be eating has benefits to their metabolic dysfunction. So I'd very much like to put these probiotic treated mice into a metabolic profiling to see if it alters that feeding regulation. But we don't know yet. Basically, I mean, you know, I guess that we haven't even collected all of the mice from our initial pilot group. You know, the first things first, we wanted to see if we give this probiotic, does it colonize the gut? Does it increase IPA? Other folks have shown that, but we'd like to see it in our hands. And does it slow weight loss in HD mice? And the answers for those things so far seem to be yes. And this is, you know, it's a reason to look deeper. And so we've got a lot of different things that we want to look at.
Well, thank you for presenting it. I think it's exciting. I mean, weight loss is a huge issue for our HD patients, and obviously a negative predictor for how they do. And a lot of people take probiotics. This is not a bacteria we see normally in probiotics, but it would be a simple thing to to do if safe?
Right. Right. And that's always the more difficult question, because safe for the general population does not always mean safe for HD. And so, you know, that's something we need to explore a little bit further, I think, in animals before we move this towards people. But that would be the eventual goal.
Thank you for joining us.
Sounds great.
Really appreciate it. Awesome talk.